Welcome to Neet-Ncert. So in this video we will see about what is the procedure of photosynthesis of the NCERT class 11 of biology. So before seeing this video, please subscribe to Neet-Ncert. If you didn't subscribe, press the bell icon at the side of it. If you like this video, like it at the end of the video. If you have any doubts or if you want more videos regarding any chapters, comment below. So we'll move into the video, guys. So for the first disclaimer is watch the video fully or else you will not find anything useful regarding this. So we'll see this photosynthesis. First, what is happening? This is PS2. This one. PS2 means P680. That is chlorophyll A containing P680. The speciality about the P680 is it only has oxygen evolving complex which can change molecule of H2O into 2 hydrogen ions and oxygen and 2 electrons. So these 2 electrons released from the splitting of the water will be taken up. Before taking up, I will tell another procedure. As the light, this one is HV, which means light. Light is entering here so that chlorophyll A gets excited into chlorophyll A plus. That is, it has lost an electron. As it has lost an electron, so it takes 1 electron from this one. 1 or 2 electrons from the electrons released by splitting of H2O. So this electron is taken up by the pheophytin, which lies, it is the primary electron acceptor, which is located towards the outer side of the, I didn't mention it, it is located at the outer side of the membrane. Outer side of the membrane transfers its electron. Now it does not, next it will transfer its electron to a compound named as plastoquinone. First I will tell you about the pheophytin. It is the primary electron acceptor of chlorophyll A P680. It is present inside the membrane, but it is present at the outer side of the membrane. That means it is present towards the stroma side of the thylakoid membrane. So next the electron moves to the compound called as plastoquinone. Plastoquinone, it is a lipid soluble quinone freely moving in the thylakoid membrane. It is not an electron carrier primarily. It is an H plus acceptor or H plus carrier. So for every electron it transports, for every electron here, electron will be here, electron it transports, it will transport on hydrogen because it is a primarily H plus transporter. For every electron transferring, it will transport the H plus from the out stroma side into H plus to lumen side. So the plastoquinone next transfers its thing into plastocyanin. Plastocyanin is a mobile electron carrier. This is an important question regarding the photosynthesis. It is a mobile electron carrier which transports its electron to cytochrome. Before coming to it, plastocyanin, here will be cytochrome B6F complex. So plastocyanin is a mediator of transporting electrons obtained from plastoquinone. From plastoquinone, the electron will move to cytochrome B6. Okay. Then the electron will move to cytochrome F. Then the electron will move to plastocyanin. This plastocyanin is a mobile electron carrier. Next, it is present in the inner side, that is lumen side of the thylakoid. Okay, it should be clear. So next, the plastocyanin will move its electron to chlorophyll A, that is P700, PS1. So P700. This PS1, the main thing is here is it does not have oxygen evolving complex, so it cannot obtain its own electron by excitation. It also gets exposed to the light, that is HV, as PS2 is also doing, but it does not have oxygen evolving complex, so it cannot split H2O into H plus. So it cannot do it. So that it only depends upon the PS2, P680 for its electron dependency. The electron obtained here moves to the chlorophyll PS1, then it moves to the ferridoxin. Ferridoxin is present on the stroma side. I will tell you about the ferridoxin. This ferridoxin, that is FD, will will move its electron to ferredoxin NADP reductase, that is FNR complex. Okay, this FNR complex will move its electron outside, which will convert NADP plus, which is electron deficient, to NADPH. Okay, this is the last procedure. This also needs H plus ion. So the next thing is, this is the process how the photosynthesis takes place. If you want to take a screenshot, take it. I will remove all these writings. So now in this thing we will see about what is the procedure of proton pump acting here. Proton pump acting here. So how it is acting? It is acting with the help of H plus ions, moving the H plus ions between them. The first thing is why H plus ions are increasing inside the lumen and decreasing outside the stroma. Three major things. That is, the first thing is, while transporting the electron from pheophytin to plastocyanin, plastoquinone is transporting H from outside stroma into lumen. That is the first thing why lumen hydrogen is increasing. Okay, and stroma hydrogen is decreasing. First point. Okay, the next point is, here hydrogen water has splitted up to form 2 H plus and half O2 and 2 electron. Okay, this has increased the H plus concentration inside the lumen. This is the second point why H plus has increased inside. The third point is this NADP plus converting to NADPH requires an electron and also requires H plus ion. So the stroma hydrogen is also decreasing. This is the third reason why stroma hydrogen is decreasing. All reasons finally causes stroma hydrogen to decrease and lumen hydrogen to increase. So there will be abundant H plus here inside the lumen, so that pH of this will be very low. What is the general thing for a movement from higher concentration to lower concentration? So H plus will get so this moving of hydrogen from inside the lumen outside to stroma creates some energy. This is how the proton pump acts. Before that, we will see about the ATP synthase, that is the proton pump here detailedly. The proton pump consists of two parts, CF0 and CF1. CF0 is embedded in the thylakoid membrane. It is embedded here. So this is CF0. And the CF1 is this one. CF1 is outside the thylakoid membrane. Sorry, not outside, it is at the outer surface of the thylakoid membrane. CF1 contains ATP synthase. As mentioned in the NCERT, if you see the diagram, it will be visible to you. The figure 13.7 of page 214. The process is, the breakdown of the gradient of H plus provides enough energy to cause conformational changes in the CF1 particle of ATP synthase, which makes the enzyme synthesis several molecules of energy packed ATP. So this movement causes production of ADP to ATP. This provides the energy. This is how ATP is produced. What are the things required for a chemiosmosis? Or a membrane, a proton pump, a proton gradient, and ATP synthase, as mentioned in the NCERT. Energy is used to pump protons across the membrane to create a gradient or a higher concentration of protons within the thylakoid lumen. This is the statement given in NCERT. So this is the final thing. Next, how the ATP produced here, how the ATP produced here and NADPH produced here, these both are energy carriers. It has energy. How these both are used are shared in the next part of the NCERT. So if you want videos regarding that, or if you want videos regarding any other chapters, please mention in the comment below. And if you found this video useful, please comment below and like the video, share to your friends so they may also get used of it. And subscribe to Neet-Ncert if you didn't subscribe till now. Thank you guys. Show you the detailed part of the video at the last also, that is this one. If you want to take a screenshot, take for it. First thing, if you want to take this one, and finally this one. This is the notes prepared by me for the video, and the other part is also there. We have making these things. So if you want, take a screenshot. Thank you guys.